Hello, let's discuss on the chemical composition of DNA. A DNA is a long polymer of nucleotide chain linked together to form a polynucleotide chain. So poly means many, so many nucleotide chain they group together to form this polynucleotide chain. So in order to understand this nucleotide, let's first discuss on the components of the nucleotide. Chemically, a nucleotide consists of three components. The first is the nitrogenous base, the second is the pentose sugar, and the third is the phosphate group or phosphoric acid. Let's first discuss on the nitrogenous base. There are two types of nitrogenous bases. The first one is purine and the second is the pyrimidine. So a purine is a double ring or dicyclic compound and is found in both DNA and RNA. There are two types of purine. The first one is adenine, represented by A. Second is the guanine, represented by G. The second type of nitrogenous base is pyrimidine. Pyrimidine is a monocyclic or a single ring compound, and there are three pyrimidine. The first one is cytosine, represented by C, and this cytosine is found in both DNA and RNA. The second is the thymine, represented by T, and this thymine is found only in DNA. The third is the uracil, represented by U, and this uracil is found only in RNA. So in case of RNA, the thymine T is replaced by U. So in RNA, instead of T, U is found, and in DNA, instead of U, T is found, that is thymine. So these are the two types of nitrogenous bases that form the nucleotide. Now the second component is the pentose sugar. Now in case of DNA, that is deoxyribose nucleic acid. The sugar, the pentose sugar that is found is the deoxyribose type of pentose sugar. In case of RNA, that is ribose nucleic acid. In case of this RNA or ribonucleic acid, the type of pentose sugar found is the ribose type of pentose sugar. Now pentose means 5. So pentose sugar, here we are referring to a sugar containing 5 carbon atoms. So 1, 2, 3, 4, 5. So this is the pentose sugar containing 5 carbon atom, and at the top it has oxygen. Now if we look at the structure of ribose and deoxyribose, it's almost very similar. In case of ribose, this is the ribose structure, ribose sugar structure. Now the first carbon atom consists of H and OH, hydroxyl group, and the second is containing H and OH. The third is also containing H and OH, and the fourth carbon is containing CH, H, and the fifth carbon is containing CH and OH. Now in case of deoxyribose, the first carbon is containing H and OH, the second carbon is containing H and H. In this case, one oxygen molecule is lost, and it is due to this reason it is called as deoxyribose. So they are all remain the same in case of ribose and deoxyribose. Now the third component is the phosphate group. Here phosphate group is also referring to the phosphoric acid, represented by H3PO4. So a nucleotide consists of a nitrogenous base, a pentose sugar, and a phosphoric acid. Now let's discuss on the types of nucleic acid. There are 4 types of nucleic acid in DNA. We are going to discuss about the nucleotide of the DNA only. The first type of nucleotide is the adenosine monophosphate. Since a nucleotide consists of a nitrogenous base, pentose sugar, and a phosphoric acid, the first nucleotide is adenosine monophosphate. That is, the first nitrogenous base in this case is a purine form of nitrogenous base, that is adenine, plus pentose sugar. Now in case of DNA, I told you the sugar is of deoxyribose type. Third is phosphoric acid, that is H3PO4. So adenine, deoxyribose, and phosphate group or phosphoric acid, when they combine, they form adenosine monophosphate. When we say mono, we are referring only to one phosphate group. So adenosine monophosphate is also represented by AMP. If there are two phosphate group, then it will be diphosphate. So adenosine diphosphate. If there is three phosphate, then it will be triphosphate. So the name will be adenosine triphosphate or ATP. Now structurally, the nitrogenous base and the phosphoric acid are attached to the pentose sugar in this form. The first carbon atom is attached to the nitrogenous base, and in this case we are taking adenine. So adenine will be attached to the first carbon atom, and the phosphoric acid, the phosphate group, will be attached to the fifth carbon atom of this pentose sugar. So this is how structurally the phosphate group and the nitrogenous base are attached with the pentose sugar. Now the second type of nucleotide is the guanosine monophosphate. Guanine plus deoxyribose sugar plus H3PO4, that is phosphoric acid. So this will give rise to guanosine monophosphate, and structurally it will be like this. It's same, but in this case, in case of guanosine monophosphate, the adenine in this case will be replaced by G. So here it will be G. So the other all remain the same. Hence this compound, this guanosine monophosphate, shows that it is containing only one phosphate. If there is two phosphate, then it will be di. Therefore the name will be guanosine diphosphate. If there is three phosphate, it will be guanosine triphosphate. Now the third type of nucleotide is the cytidine monophosphate. So cytidine here refers to the presence of cytosine, nitrogenous base, with deoxyribose and phosphoric acid. In this case also it's same, only in this case the nitrogenous base, instead of A, it will be cytosine. So cytosine will be here C. So the additions of this phosphate with pentose sugar and cytosine nitrogenous base will give rise to cytidine monophosphate. That means one phosphate. If there is two, it will be cytidine diphosphate. If it is three, it will be cytidine triphosphate. Now the fourth type of nucleotide is the thymidine monophosphate. In this case, thymidine here refers to presence of thymine nitrogenous base with deoxyribose and the phosphate or the phosphoric acid. In this case, instead of adenine, it will be thymine. So this will give rise to thymidine monophosphate or TMP. If it is 2 phosphate, means thymidine diphosphate. If it is 3 phosphate, means thymidine triphosphate. So this nucleotide with deoxyribose sugar is called as the deoxyribose nucleotide. So structurally, the nucleotide will be arranged in this form. The first this is the first nucleotide, second nucleotide, and the third nucleotide. Nucleotide means nitrogenous base, pentose sugar, and the phosphoric acid. So the presence of all these three give rise to the nucleotide. So the first nucleotide will be attached to the second nucleotide through the third carbon atom of the first one and the fifth carbon of the second nucleotide. So in this case, the first nucleotide is attached to the second nucleotide from the third carbon atom, and the second nucleotide is attached to the first from the fifth carbon atom. So the attachment of this third and the fifth give rise to a type of bond called as the phosphodiester bond. So phosphodiester bond is the bond that exists between the two nucleotides. So two nucleotides are joined by a type of bond called as the phosphodiester bond, and the phosphate, the pentose sugar is attached to the nitrogenous base with a type of bond, this bond called as the glycosidic bond. So glycosidic bond exists between the nitrogenous base and the pentose sugar. Now the third nucleotide is attached to the second in the same way. The third carbon atom of the second is attached to the third through the third carbon atom, and the third nucleotide is attached to the second nucleotide from the fifth carbon atom. So this bond that exists here is called as the phosphodiester bond. So if we look at the attachment, here the fifth is attached to the third carbon atom. The fifth carbon atom of this is attached to the third carbon atom. Here also the fifth carbon atom of this is attached to the third carbon atom of the first one. Therefore, the direction of attachment is called as the 5 towards 3. 5, 3. So this arrangement is called as in the direction of 5 dash to 3 dash. Now, the attachment of this 1, 2, 3 till 20 is called as the oligonucleotide, and the attachment of this 1, 2, 3 and above 20 nucleotide is called as the polynucleotide. And this polynucleotide, they link together to form nucleic acid. So in case of DNA, it's called as deoxyribose nucleic acid with ribose deoxyribose sugar. In case of RNA, it's called as ribonucleic acid with ribose type of pentose sugar.